and welcome everybody to my channel Omini Bio Entrance, and today we will be discussing the Telangana SET question paper for the subject Life Science. I am discussing the paper 2 section, and this was conducted in the year 2024. So coming to the first question, which of the following statements are correct? Polarity of water molecule makes it an excellent solvent. That is correct. And water is having polarity means it means that water is having a hydrogen and oxygen atoms. So oxygen is having a partial negative charge and hydrogen is having a partial positive charge. So that is what is meant by polarity. Because of this polarity, water can attract other polar molecules and dissolve them in water. Okay, so it makes an excellent solvent. That is correct. Water has a high tensile strength. Now water molecules, they are attached to one another because of the cohesive force, that is between the same molecules, force of attraction between same molecule. So water is having H2O, 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 they are attracted to each other. Now this allows the water to resist being pulled apart. Okay, so it is having high tensile strength. Then cohesive property of water, yes, that is also correct. Water has high dielectric constant. We are learning it from our +2 onwards. So that is also correct. So 1, 2, 3, 4 are correct. That makes option 4 as the answer. Then next one, the peptide bond in a protein is. So peptide bond rotates to 3 preferred dihedral angles. Now the peptide bond, it is not able to rotate freely. Okay. Now it is having a partial double bond character because of the now CONH bond. This is the peptide bond between two amino groups. Now it is not able to rotate freely. Okay, and it has got partial double bond character because of resonance, where the pi electrons are shared between the carbonyl oxygen and the amide group of the nitrogen. So because of this sharing, the peptide bond, it is rigid and it is planar. Okay, and these CONH, they are always in the trans configuration. That means these groups, these R groups, they are on opposite side of the peptide bond. That is, one R group is on this side, means of one amino acid will be here, the R group will be on top here, this side, not down. Okay, so the R groups, they are on opposite side. That is why the peptide bond is having a trans configuration, and it minimizes the steric hindrance caused by these bulky R groups of amino acids. Okay, and but rotates to 3 preferred dihedral angles. Okay, it is planar, but it doesn't rotate freely due to the partial double bond character. Now between the N and C alpha, you have got the phi dihedral angle, and between C alpha and C you have got the psi, and for the peptide bond there is the omega 1. So the restrictions, they are this rotation of these phi, psi, etc. angles, they are restricted to only specific angles, not to full rotation is not there. Now that is to prevent steric clashes between the R groups of various side chains of amino acids. And these combinations of this phi and psi dihedral angles can be visualized using the Ramachandran plot. Now next question is for Ramachandran plot, I think. Then non-planar, that is not correct. Planar and usually found in trans configuration. Yes, peptide bond is planar and it is usually found in trans configuration, that is at 180 degrees. Now next question, choose the correct statement about peptides in the Ramachandran plot. Now the Ramachandran plot, it provides a overview of the allowed and disallowed regions of these based on the dihedral angles. Okay. Now you can also see the plot and predict the structure of secondary structure of the protein. So you can find the beta helixes, beta sheets, alpha helices in the Ramachandran plot. Okay, now come to the options. Peptides that are unstructured. Now unstructured peptide means they lack the stable fixed three-dimensional structure. They are usually flexible, disordered state and are usually called as random coils. Will have all the backbone dihedral angle in the disallowed region. No, it is in the allowed region. The disallowed region of the Ramachandran plot is mainly for the mainly indicate the steric clashes which arise in peptides. So that is what the disallowed region is showing. But random coils, they are naturally occurring. So there are no they won't be found in the disallowed region, but they will be found in the allowed regions. Only thing, they will not have proper structure, just like the alpha helix, beta sheet, beta turns, etc. Then it is not possible to conclude whether peptide adopts helix or beta sheet conformation. You can conclude or predict the structure of the peptide, whether it is alpha helix or beta sheet conformation based on the Ramachandran plot. Then the occurrence of beta turn conformation in a peptide. Yes, beta turns can be detected in Ramachandran plot. If you know the plot, you can find that the beta turns are usually confined to the upper right quadrant. That is where you find your left-handed alpha helices. In that quadrant also you can find beta turns. Then sequence of peptide can be deduced. And the sequence of the peptide cannot be deduced with the help of Ramachandran plot. It only tells regarding the conformation the peptide can adopt, whether it is going to have an alpha helix or a beta sheet structure, not the sequence. Then next one, amino acid tend to be least soluble in water at their isoelectric point. Yes. So at their isoelectric point means it is the pH where the amino acid will not have a no net charge. So the net charge is 0 on the amino acid. So it cannot interact with the water, and it will be less soluble in water, and it will precipitate out in the solution. Okay. So that is why amino acid are least soluble in water at their isoelectric point. Now what is the reason they have given? The net charge on an amino acid is a function of the pH of the solution. Of course that statement is correct. At a low pH the amino acid gains protons and it is positively charged, and at high pH the amino acid will lose the protons and become negatively charged. So whether the amino acid is positively charged or negatively charged, it is based on the pH of the solution. But that is not the reason why amino acid tend to be less soluble in water at isoelectric point. Now why are they not soluble? Because at isoelectric point the amino acid doesn't have a charge. So they cannot effectively interact with the water molecules, making them less soluble and precipitate out of the solution. So here the answer will be both assertion and reason are true, and the reason is not the correct explanation of assertion. So that is correct. Then which of the following statement about B DNA structure is incorrect? B DNA is what the Watson and Crick model describes. So within a double helix there are 10 bases per turn of the helix, of course. The double helix contain anti-parallel chains, yes, one in 5' 3' direction, other in the 3' to 5' direction. So anti-parallel, that is also correct. The amount of adenine plus thymine equal amount of guanine plus cytosine, that is not correct. It is actually the sum of purines is equal to sum of pyrimidines. Now what are purines? Adenine and guanine. So A plus G equals to sum of purines, that is pyrimidines, that is thymine and cytosine. So T plus C. So what they have given? Adenine plus thymine. A plus T is equal to G plus C, which is wrong. It should be A plus G equal to C plus T. That is your Chargaff's rule. So 3 is incorrect. Then separation of the two strands of the double helix require untwisting of the helix, yes, by topoisomerase, helicases, etc. So 4 is correct. Then glucose and mannose are epimers. This means, now epimers means they have more than one chiral carbon atom, but they differ from each other in the configuration of only one chiral carbon atom. Chiral means the carbon atom having different groups attached to it. So this carbon atom, if you take, it is having the aldehyde group here, OH group here, hydrogen here, and an entirely different group over here. Similarly for mannose also, these groups are 4 different groups are attached to the carbon atom. If you take this one, a different group is attached here, another here, another here, another here. So if you take this one, again a different group is attached to all the 4 valencies of the carbon atom. Now glucose and mannose are epimers, means they differ in the configuration at only one chiral carbon atom. So here that chiral carbon atom is at position 2. So C2 carbon, they are different. Okay, so they are epimers. They are mirror images? No. Here OH is on this side and in mannose OH is on this side. How will they be mirror images? No. One is aldose, other is ketose. No, they both are aldose. See here. So that's also not correct. They rotate the plane polarized light in opposite direction, that is with regard to your enantiomers, not epimers. They differ only in the configuration of one carbon atom, yes, C2 at position 2. So that statement is correct. So 4 is the answer. Now which of the following statements are correct? Double bonds in unsaturated fatty acids are generally in the cis configuration. Yes, that is correct. They are in the cis configuration. Unsaturated means they are having double bonds. Then in polyunsaturated fatty acid, double bonds are almost never conjugated. Now what is this conjugated fatty acid mean? So a conjugated fatty acid have a chain of alternating single and double bonds. So double bond, single bond, double bond, single bond, double bond, single bond. That is conjugated fatty acid. Now non-conjugated means they have double bonds which are separated by at least one methylene group. See here. So double bond here, both are separated by a methylene group, that is unconjugated fatty acid. Now what are they telling? In polyunsaturated fatty acid, double bonds are almost never conjugated. Yes, that is correct in case of the PUFAs. Then sphingoglycolipids are involved in biological recognition. Yes, sphingoglycolipids, they are found on the outer leaflet of the plasma membrane. They have the carbohydrate group which is exposed outside, and this carbohydrate group, it will help in cell to cell communication, then adhesion, as well as in immune responses. So 3 is also correct. Prostaglandins contain a 5-membered ring of carbon atoms. That is also correct. Prostaglandins regulate the synthesis of intracellular messenger CAMP, which is not correct. Of course, prostaglandins are involved in signaling pathways. They are involved in inflammation, immune responses, but they do not regulate the synthesis of CAMP. CAMP, we found that it is mainly with regards to GPCR signaling or G protein coupled receptors. So that is not the right answer. So 5 is wrong. Then Michaelis-Menten equation state rate of enzyme reaction is independent of substrate concentration. That is correct. See here, this is the rate of reaction, this is substrate concentration. Substrate concentration increases, rate also increases, and there is a stage where it reaches the Vmax, which state that your enzyme is fully saturated with the substrate. There is no single enzyme molecule that is not bound to the substrate. At that time, it reaches the what is called as maximum velocity. Okay, so substrate concentration increases, rate of reaction increases in the beginning, but as the substrate concentration become more and more, it reaches the Vmax. So rate of enzyme reaction is independent of substrate concentration is not correct. Then rate of non-enzyme reaction, Michaelis-Menten, they have asked in the question, Michaelis-Menten equation. It is for enzyme catalyzed reaction, not for non-enzyme reaction. Then Km is the enzyme substrate complex association constant. No, it just tells us the concentration of substrate that is needed for the reaction to reach half Vmax. That is what is meant by Km. Okay, the substrate concentration required to reach half the maximum velocity. So a low Km value means the enzyme requires only a low substrate concentration to reach half Vmax. That is, it has more affinity for the substrate. So low Km, high affinity for the substrate. Okay, and the low Km, it also suggests that the complex, the enzyme substrate complex is not going to dissociate at a very faster rate. Then enzyme, the fourth one, enzyme substrate complex formation is essential in enzyme reaction. Yes, enzyme substrate complex is required for enzyme reaction to occur, because Michaelis-Menten kinetic, it describes the formation and breakdown of this enzyme substrate complexes. So here as the substrate concentration increases, enzyme substrate complex formation is also increasing. And at higher substrate concentration, all the enzyme molecules are complexed with their substrate. Okay, so fourth is the answer. Then which of the following statements are correct about retrotransposons? So retrotransposons, they are also like your transposons. They are mobile genetic elements, but they transpose themselves via an RNA intermediate. That is by copy paste mechanism. So this is your DNA having the retrotransposon, it will use the RNA polymerase of the host, create a copy of the RNA, then it will use its reverse transcriptase. That is, retrotransposon is having a reverse transcriptase gene in it, so it will use that one, and it will construct the cDNA, integrate the cDNA into the chromosome. This is the chromosome, and the retrotransposon has integrated into a new region of the chromosome. So this mechanism is copy-paste mechanism of retrotransposon movement. Then they transpose via RNA intermediate, of course. They contain genes for ribosomal proteins. They do not contain such genes. They possess a gene for RNA-dependent RNA polymerase. No, they have the reverse transcriptase which convert RNA to DNA. RNA-dependent RNA polymerase, it will synthesize RNA from an RNA template, and it is found in viruses. Here it is using the RNA polymerase of the host, not its own RNA polymerase. So it doesn't have that gene. They are found only in bacteria. No, they are found in archaea, eukaryotes also. They may contain introns. Yes, that is also correct. During their evolution, they must have had introns. So 1 and 5 are correct. Then which of the following accurately describes the Na plus K plus ATPase pump role in cellular physiology? So Na plus K plus pump, it pumps the 3 Na positive out of the cell and 2 K plus into the cell. So and ATP is consumed in the process, and this is against the concentration gradient. Okay, so facilitate passive diffusion. Passive diffusion does not use ATP, but here Na plus K plus it use ATP, so 1 is not correct. Use ATP to pump ions against gradients. Yes, that is correct. Regulates water movement. Okay, now that one, it is not a direct function of Na plus K plus ATPs. Now what it is doing? It is transporting Na plus out of the cell. That is what Na plus K plus ATPase is doing. Now what will happen if there was excess Na plus in your cell? So if excess Na plus is there in your cell, more ion concentration inside the cell, water content inside the cell is low. So what will happen? Water will enter into the cell from the surroundings. So excess water influx. Now because of this excess water influx, the cell will swell and it will lead to cell lysis. Now what is Na plus K plus ATPase doing? It is transporting Na positive out of the cell, thereby preventing accumulation of Na plus inside the cell. Okay, so that is what Na plus K plus ATPase pump is doing. So it is maintaining the osmotic balance and cell volume. So it is an indirect role, not a direct one. Here they have asked accurately describes. So that is just an indirect role. So we will not take that into consideration. Main accurate answer is uses ATP to pump ions against gradients. Operates independently of energy. No, it requires energy. That is in the form of ATP. Then which of the following accurately describe the mechanism by which bacteria respond to oxidative stress? So activation of heat shock protein to repair damaged DNA. Yeah, that is correct. For activation of mechanism to repair damaged DNA. So A1, that is correct. Up regulation of antioxidant enzymes to neutralize reactive oxygen species. Yes. See, because of this it is oxidative stress, so reactive oxygen species are present in more quantity. So the bacteria will produce antioxidant enzymes, that is like superoxide dismutase, that will convert the superoxide anion, that is O2- minus into H2O2 and O2. Then there is the production of catalase and peroxidase, which will break down the H2O2 into H2O and oxygen. So that is correct. Inhibition of protein synthesis to conserve energy. This is for reduction in metabolic activity to save resources. Yes, the bacteria is reducing its metabolic activity and it will direct its energy towards the detoxification action as well as in repair to damaged DNA. So C is 3, so that's also correct. Increased production of osmoprotectant to maintain cell membrane integrity. Yes, it will produce. Use the trihalose, the glycine, betaine, all these things as osmoprotectants and maintain cell membrane integrity, stabilize what is that protein and prevent aggregation. So strengthening of cell structures to withstand environmental stress. So D4. So 1 is the answer. Then next one, peroxisomes play a crucial role in detoxification reaction, particularly in the breakdown of hydrogen peroxide. Yes, they are having catalase breaking down hydrogen peroxide. So that is correct. Their main function is to store calcium ions and regulate intracellular calcium levels. No, peroxisomes are not involved in storing calcium ions. Calcium ion storage is by your endoplasmic reticulum. If you remember your muscle contraction, the smooth endoplasmic reticulum has released the calcium out into the cell from its lumen. Okay. So the main function of calcium storage is for endoplasmic reticulum, not for peroxisome. So the answer is, the assertion is correct, but the reason is incorrect. Then which of the following accurately describe the function of anaphase promoting complex cyclosome APCC in the regulation of cell cycle? Now the anaphase promoting complex, it is a E3 ubiquitin ligase. That is, it will add a ubiquitin molecule to the target protein. So by adding ubiquitin molecules, it is directed to proteasome degradation. Okay. Now the main function what I have seen in various textbook is metaphase to anaphase transition. That is, okay, before that this anaphase promoting complex, it has got two co-activators, that is CDC20 and the CDH1. So if it is metaphase to anaphase transition, then this APC, it will be bound by the co-activator CDC20. And what will this do? It will metaphase to anaphase means there should be separation of the sister chromatids so that they move towards the opposite poles. So you know the sister chromatids, they are held together by the cohesin protein. Now what does APC CDC20 it will do? It will ubiquitinate the securin and the mitotic cyclins. Okay. Now securin will be degraded upon ubiquitination. So the securin has caught hold of a separase and kept it with itself. Now once the securin is degraded, separase is becoming free. And this separase, what it will do? It will cleave the cohesin that is holding the sister chromatids and they separate during anaphase. That is what APC is doing from metaphase to anaphase transition. Now they have told APC initiates a dash during the S phase. It has no role in DNA replication based on whatever articles I have read. So I am ignorant about that if it is having any role also. As per me, it is not there. Okay. Now next role of anaphase promoting complex is mitotic exit and G1 entry. Now after completion of the mitosis, the cell shouldn't go and start another round of mitosis. It has to take rest. Why? That is, it has to enter the G1 phase. That is for the production of certain growth factors and enzymes which are needed for the next round of the cell cycle. Okay, now this is done by inhibiting the CDK activity. Now CDK is a phosphorylating protein, and its activity is CDK becomes active upon binding to cyclins. Now what does APC do here? APC with its coactivator CDH1 here, it is for CDH1, I told you it is for mitotic exit and G1 entry. It will prevent the accumulation of cyclins that will trigger the next round of mitosis. So what the cell will be forced to do, it will have to stop, it will have to exist the mitosis and enter into the G1 phase of the cell cycle. Okay. So APCC degrades cyclins and other regulatory protein to dash through the cell cycle. It is allowed progression through the cell cycle. So metaphase to anaphase progression is there, and from mitotic exit to G1 phase progression. So this portion is correct. Then what is the function of an operator in an operon system? So operator, it is a DNA sequence to which your repressor will bind. Now once the repressor is bound to operator, it will block RNA polymerase from binding to the promoter. So RNA polymerase not bound, the operon structural proteins will not be synthesized. Now if the repressor is released from the operator, then RNA polymerase can bind to the promoter, thereby turning on the structural genes. So it is just like a switch. It is a switching off the expression of structural genes or switching on the expression of structural genes. So it serves as a site where RNA polymerase binds. No, RNA polymerase bind to promoter, not to operator. It codes for structural genes. No, if you see the lac operon, it is coded by the lac ZYA genes, which are not part of the operator. Then it acts as a switch, yes, controlling the access of RNA polymerase to the genes in the operon. 100% correct. So 3 is the answer. It is responsible for the synthesis of a regulatory protein that control gene expression. No. Then next one. During which stage of prophase 1 in meiosis does synapses occur leading to the formation of tetrads? So here if you see, you have got the leptotin, zygotin, pachytin, diplotin, diakinesis, 5 stages of meiosis 1 or prophase of meiosis 1. So first leptotin, that is the duplicated chromosome, they start to condense. Then the homologous chromosome pair and synapse, thereby forming the synaptonemal complex. So zygotin, they are the tetrads. Then pachytin, synapse is complete, and then crossing over take place between the non-sister chromatids of homologous chromosomes. Then the diplotin, the synaptonemal complex will start disappearing, but they remain attached at the points called as chiasmata. Then at diakinesis, the chromosomes are maximally condensed, nuclear envelope breakdown, spindle fibers begin to form, bivalent are ready for the metaphase. So here they have asked, does synapses occur, that is in your zygotin phase, and formation of tetrads. So the answer is option 2, that is zygotin. Then how transposons are typically arranged in a genome? They are just scattered in the genome. So within exons, no, they are found also in exons or in interrupting coding sequence. Exclusively in promoter, no. Scattered throughout the genome, including within genes, intergenic regions, yes. Confined to specific transposon clusters, no.